Nature has a remedy for the problems we create. Assalamu alaikum wa rahmatullahi wa barakatuh. I am Muhammad Ashfaq of Grade 10, here to present on a wonderful research project, isolation of halophiles and their application towards degrading oils. First of all, what are halophiles? Halophiles are defined as salt-loving organisms. These are found in extreme places like oceans, salt lakes, and seas. The aim of this project is to isolate these halophilic bacteria and biodegrade non-biodegradable items in the oil. So, why do we need to degrade oils? Oil spillage has been the main problem for the past decade. Around 700 million tons of oil was spilled only in 2021, even though the export declined due to COVID. We need to degrade these oil contents. So what is the aspect and impact of this project? Aspect is that spillage or releasing of hazardous oil in the marine environment and impact destruction of ecosystem of marine. The plan for experimental design. First of all, we are going to isolate these halophilic bacteria from salty environment like oceans, and then we are going to apply them on oils. So first, the research questions are, how can we isolate these halophilic bacteria? Will halophiles help in degradation of oils? If so, can it degrade all types of oils? The hypothesis of the project is halophilic bacteria will help in biodegradation of hazardous oil contents. The methods of this project. The methods of the project includes 5 stages. The first one is preparation of nutrient agar. The second one is cultivation of halophiles from samples. The third one is isolation and streaking of the obtained colony. The fourth one is preparation of nutrient broth and acclimatizing the culture with oil. And the fifth stage is application of broth and colony on oil samples. The first stage of the project is preparation of nutrient agar. For this, we would require 14 grams of agar powder and 500 ml of distilled water. First, we have to mix these together and keep these in autoclave with all other equipments like petri plates and micropipette in the autoclave for 15 minutes until it reaches 121 degrees Celsius in 15 pounds pressure. The second stage of the project is cultivation of halophiles from samples. The samples are collected from ocean water and we are going to spread these in the petri plates with micropipette. After spreading these, we are going to add nutrient agar and wait it until it solidifies. After it solidifies, we have to invert it and keep it in the incubator at 37 degrees Celsius for 5 days. After incubating, we would see the bacterial colony as shown. The first two pictures shows the first bacterial colony. After this, we are subculturing it for 15 days interval for 5 times to obtain the pure halophilic colony. The next stage of the project is applying these oils on the halophilic colony. For this, we would require 3 oils: vegetable oil, diesel and engine oil. Before that, we have to prepare the nutrient broth. To prepare nutrient broth, we would require 6.5 gram of nutrient broth powder and 500 ml of distilled water. After adding these together, we have to incubate these along with the oil. We have to sterilize these along with the oil in the autoclave for 15 minutes until it reaches 121 degrees Celsius. After incubating, we have to add 5 ml of oil in this broth along with the colony and incubate it for 1 to 2 days. This is the incubation pictures. And the last stage of the project is to apply the oil samples and colony on broth. After the incubation of these acclimatized culture, we are going to add 16 ml of oil and 50 ml of broth into that culture. And we are going to incubate it for 5 days for the bacteria to act on the oil. After finishing all the procedures, we came on to the results. First, we check the absorbance level of oil sample without colony. When checking the oil samples without colony, we got in vegetable oil around 0.713, in diesel about 0.956 and in engine oil about 0.918. And when we check the oil sample with colony, in vegetable oil we got about 0.693, in diesel you got about 0.887, and in engine oil you got about 0.869. From these two tables, we can conclude that diesel has reduced the most. To ensure that the hazardous oil contents has been reduced, we went on to test nitrate and sulfur. First, we are doing the nitrate test. For nitrate test, we used alpha-naphthalamine and sulfonic acid, and we added 5 ml of each oil sample in these. The one which turns red shows that the nitrate has been reduced. In all the 3 oil samples, we saw the color turning red. After 3 minutes, the color started turning, and after 5 minutes, we could see pure red in it. In vegetable oil, we found the color as reddish orange, and in engine oil, we found the color was dark red, and in the diesel, we found the color was a medium red. From this, we can say that in engine oil, the nitrate content has been degraded the most. And then we tested the sulfur. We know that sulfur produces pungent smell. We can see the bubbles of H2S gas too, and when we opened it, we found a pungent smell. From this, we can say that the bacteria has degraded hazardous oil contents like nitrate and sulfur. Now, what is the application of this project? This research can be applied in many places of oil spillage, especially in marine environment. Like, there was about 700 million tons of oil spillage only in 2021. If we apply these over those areas, we can reduce the hazardous oil contents, save the environment. Now, coming on to the conclusion of the project. Now, we are checking whether we have obtained the result of the research questions. First research question was, how can we isolate halophiles? We have successfully isolated the halophiles and we have applied them on oil samples. The second question was, will halophiles help in degradation of oils? Yeah, we have used these halophiles and degraded the oil contents, and we have even checked the absorbance level, did the nitrate test and the sulfur test. And the third question was, can halophiles degrade all types of oils? We have degraded engine oil, vegetable oil and diesel. So, it may degrade all types of oils. The future enhancement of this project is to research the feasibility of the application in real life. We are going to use crude oil and whether we could degrade these hazardous oil contents. The aim of the future enhancement is to convert the halophiles into a spray, like how choppers used to spray the fire extinguishers on forest fires. Like that, we are going to convert these halophiles into a solution which could be sprayed by the choppers. Jazakallah khair. Assalamu alaikum wa rahmatullahi wa barakatuh.